Hello everyone, Professor Beda. Let's talk about cellular respiration and fermentation. We need energy to do literally anything from running a race to simply breathing, and every cell in your body is furiously producing all this energy all the time. So, where does this energy come from, or how is the chemical energy stored in uh, food used to generate ATP, the molecule that drives most cellular work? Living cells requires transmission of energy from outside sources to perform their many tasks. For example, assembling polymers, pumping substances across membrane, moving and reproducing. The outside source of energy is food, and the energy stored in organic molecules of food ultimately comes from the sun. Energy flows into an ecosystem as sunlight and exits as heat. In contrast, the chemical elements essential to life are recycled. Photosynthesis generates oxygen and as well as organic molecules used by mitochondria of eukaryotic cells and uh, the mitochondria as a fuel for cellular respiration. Uh, Respiration breaks this fuel down and using oxygen and generating ATP. The waste product of this type of respiration is carbon dioxide and water. Those represent the raw materials for photosynthesis. Let's consider how cells harvest the chemical energy stored in organic molecules and use it to generate ATP, the molecule that drives most cellular work. Uh, so, metabolism or metabolic pathways that release stored energy by breaking down complex molecules are called catabolic pathway. Transfer electron from food molecules like glucose to another molecule plays a major role in these pathways. Organic compounds possess potential energy as a result of the arrangement of electrons in the bond between their atoms. The cell degrades complex organic molecules that are rich in potential energy to simpler waste products that have less energy. Some of the energy taken out of chemical storage can be used to do work. The rest is dissipates as heat. One catabolic process, fermentation, is a partial degradation of sugar or other organic fuel that occurs without the use of oxygen. And however, the most efficient catabolic pathways is aerobic respiration, in which oxygen is consumed as a reactant along with the organic fuel. The cells of most eukaryotic and many prokaryotic cells or organisms can carry out aerobic respiration. Some prokaryotes use substances other than oxygen as a reactant in similar process that harvests chemical energy without oxygen. This process called anaerobic respiration. So the term cellular respiration includes both aerobic and anaerobic processes. However, it originated as synonym for aerobic respiration because of the relationship of that process to organismal respiration, in which an animal breathes in oxygen. Thus, cellular respiration is often referred to the aerobic respiration. So we have an organic compound. Those are carbohydrates, fat, proteins, um, in animal diet. Um, the major source of carbohydrate is starch, and you know that starch is storage polysaccharide that can be broken down into glucose, C6H12O6 subunit. Here we will learn the steps of cellular respiration by tracking the degradation of the sugar glucose. Uh, in the presence, it will break down with the presence of oxygen and the mitochondria and release carbon dioxide, water. Those are the raw material for photosynthesis that the plant absorb them from the atmosphere and from soil. And the breakdown of glucose uh, will produce energy in the form of ATP, and the other energy is dissipated as heat. The breakdown of glucose is exergonic reaction. So recall that a negative delta G, that means delta G less than 0, indicates that the products of the chemical process store less energy than the reactants, and that the reaction can happen spontaneously, in other words, without an input of energy. To keep working, the cell must regenerate its supply of ATP from ADP, adenine diphosphate, and inorganic phosphate. To understand how cellular respiration accomplishes this, let's examine the fundamental chemical process known as oxidation and reduction. How do the catabolic pathways that decompose glucose and other organic fuels yield energy? The answer is based on the transfer of electrons during chemical reaction, and the relocation of electrons release energy stored in organic molecules, and this energy ultimately is used to synthesize ATP. In many chemical reactions, there is transfer of one or more electrons from one reactant to another. These electron transfers are called oxidation reaction or redox reaction for short. In redox reaction, the loss of electrons from one substrate or one substance is called oxidation. The loss of electron is oxidation, and the addition of electrons to another substance is known as reduction. Note that adding electrons is called reduction. Adding negatively charged electrons to an atom reduces the amount of positive charge of that atom. To take a simple non-biological example, consider the reaction between the element sodium and chlorine that form table salt and ACl. You see that sodium becomes oxidized, that means it loses its electron and becomes positive, and chlorine becomes reduced, that means it gains electron. So we can generalize the reaction of or redox reaction, uh, substances X and Y. This one is XY. This substance is electron donor, and we call them reducing agent. It reduces Y. This is a reducing agent. Okay, the electron donor, the same reducing agent. It will reduce substance Y. Substance Y accepts the donated electron, so the electron acceptor, we call it the electron acceptor. So Y is the oxidizing agent. It oxidizes X by removing its electron. Okay, because an electron transfer requires both an electron donor and an acceptor, oxidation and reduction always go hand in hand. Not all redox reactions involve the complete transfer of electron, like this generalized reaction. That means not all those reactions involve the completely transfer of electron from one substance to another. Some change the degree of electron sharing in covalent bond. In this example, the covalent electrons is methane are shared nearly equally between the bonded atoms, between the carbon and hydrogen atoms. Because carbon and hydrogen have about the same affinity for valence electrons, they are about equally electronegative. But when methane reacts with oxygen, forming carbon dioxide, electrons end up shared less equally between the carbon and its new covalent partners, the oxygen atom, which are very electronegative in effect. So the carbon atoms has partially lost its shared electrons. Thus, methane has been oxidized. Uh, so electron sharing in covalent bond shown here for each reactant and product. So on methane, the reducing agent carbon shares a pair of electrons equally in 4 covalent bonds with each of 4 hydrogen atoms, and in oxygen, the oxidizing agent, so the oxygen is the oxidizing agent, 2 pairs of electrons are equally shared in a covalent bond, uh, are equally shared in this between the oxygen. In carbon dioxide, carbon shares 2 pairs of electrons with each of 2 oxygen atoms, but the electrons are not shared equally and are pulled toward both oxygen atoms. In water, oxygen share a pair of electrons with each of 2 hydrogen atoms, but the electrons are not shared equally and are pulled toward oxygen. So oxygen is electronegative, and this is an example of non-completely transfer of electrons from one substance to another. The oxidation of methane by oxygen is the main combustion reaction that occurs at the burner of a gas stove. The combustion of gasoline in automobile engine is also a redox reaction. The energy released pushes the pistons. The oxidation of glucose and other molecule in food. Let's examine again the summary equation for cellular respiration, but this time think of it as a redox process. The fuel, glucose, is oxidized, become oxidized, and oxygen is reduced. The electrons lost potential energy along the way, and energy is released. In general, organic molecules that have abundance of hydrogen are excellent fuel because their bonds are a source of electrons, whose energy may be released. These electrons fall down in energy gradient during the transfer to oxygen. In respiration, the oxidation of glucose transfers electrons to a lower energy state, liberating energy that becomes available for ATP synthesis. So in general, we see fuels with multiple carbon hydrogen bonds oxidized into, into products with multiple carbon and oxygen bonds. Cellular respiration doesn't oxidize glucose or any other organic fuel in a single exclusive step. but glucose is broken down in a series of steps, each one catalyzed by an enzyme. At key steps, electrons are stripped from the glucose. In oxidation reaction, each electron travels with a proton, and thus it travels in the form of hydrogen atom. The hydrogen atoms are not transferred directly to oxygen, but instead are usually passed first to an electron carrier or coenzyme called nicotinamide adenine dinucleotide. This one is a derivative of vitamin niacin, vitamin B3. This coenzyme is well suited as an electron carrier because it can cycle easily between its oxidized form, NAD, and its reduced form, NADH. NAD, as an electron acceptor, function as an oxidizing agent during respiration. Uh, how does NAD trap electrons from glucose and other organic molecules in food? Enzymes called dehydrogenase removes a pair of hydrogen atoms, 2 electrons and 2 protons, from the substrate. The substrate may be glucose or any organic molecules, thereby oxidizing it. Okay? The enzyme delivers the 2 electrons along with 1 proton to its coenzyme, NAD, forming an ADH. The other proton is released as a hydrogen atom or ion into the surrounding solution. So the full name of NAD is nicotinamide adenine dinucleotide. The molecule consists of 2 nucleotides joined together at their phosphate groups, shown here in yellow. Nicotinamide is nitrogen base. The enzymatic transfer of 2 electrons and 2 hydrogen or 2 proton from the hydrogen or from the organic molecules in food to an AD. Okay, and then the NAD is reduced to an ADH. Most of the electron removed from food are transferred initially to an AD, forming an ADH. So this is reversible reaction with enzyme dehydrogenase. To the right, reduction of NAD. To the left, oxidation of NADH. Reaction is NAD and 2 hydrogen from food, 2 hydrogen from food. Equilibrium arrow to NAD and H. The 2 hydrogen atom from food represent 2 electrons and 2 hydrogen. 1 hydrogen plus 2 electrons, 2 electrons and 1 proton will transfer to NAD and form an ADH. And the other hydrogen atom or ion released to the surrounding. How do electrons that are extracted from glucose and stored as potential energy in NADH finally reach oxygen? An electron transport chain consists of a number of molecules, mostly proteins, built into the inner membrane of the mitochondrial of eukaryotic cells. Electrons removed from glucose are shuttled by NADH to the top energy or higher energy end of the chain. And then at the bottom, we have lower energy end, the oxygen acceptor. These electrons along with the hydrogen nuclei and form water with the oxygen. The one-step exergonic reaction of hydrogen with oxygen to form water release a large amount of energy in the form of heat and light. This one is an explosive release energy. In cellular respiration, the same reaction occurs in stages. An electron transport chain breaks the fall of electrons in this reaction into a series of smaller steps and stores some of the release energy in a form that can be used to make ATP. The rest of energy is released as heat. In summary, during cellular respiration, most electrons travel the following downhill route. That means from glucose to NADH, the electron transfer from glucose to NADH, and then to electron transport chain, and finally to oxygen that's from water. The cell uses the energy released from the exergonic electron fall to regenerate its supply of ATP. So in part A, we have an uncontrolled reaction, the one-step exergonic reaction of hydrogen with oxygen to form water releases large amount of energy in the form of heat and light. That's mean in explosion. Free energy, delta G, increases in opposition to the one-step reaction, resulting in explosive releases of heat and light energy. Part B, in cellular respiration, the same reaction occurs in stages. An electron transport chain breaks the fall of electrons in this reaction into smaller steps and stores some of the released energy in a form that can be used to make ATP. The rest of energy is released as heat, and free energy increases in opposition to the stepwise progress the reaction, controlling the release of energy for the synthesis of ATP. Two electrons coming from food via an ADH are passed through a series of controlled steps in the electron transport chain. At the bottom of the chain, the two electrons and two hydrogen atom combine with oxygen to form water. In this process, ATP is synthesized. The harvesting of energy from glucose by cellular respiration is a cumulative function of three metabolic stages. Biochemistry usually reserve the term cellular respiration for the stage 2 and 3 together, but the glycolysis is part of it because most respiring cells deriving energy from glucose use glycolysis to produce the starting material for the citric acid cycle. The diagram shows glycolysis and then pyruvate oxidation and the citric acid cycle are the catabolic pathways that break down glucose and other organic fuels. Glycolysis, which occurs in the cytosol, begins with the degradation process by breaking down glucose into two molecules called pyruvate. In eukaryotes, pyruvate enters the mitochondrion and is oxidized to a compound called acetyl-CoA, which enters the citric acid cycle. Citric acid cycle here or in this cycle, the breakdown of glucose occurs to 2 carbon dioxide is completed to break down glucose to carbon dioxide. In prokaryotes, these processes take place in cytosol. Some of the steps of glycolysis and citric acid cycle are redox reactions, in which dehydrogenase transfers electrons from substrate to NAD or the related electron carrier, FAD, forming NADH or FADH2. In the third stage of respiration, the electron transport chain uh, accepts electron from NADH or FADH2 generated during the first two stages and passes the electrons or these electrons down the chain. At the end of the chain, the electrons are combined with molecular oxygen and hydrogen ion forming water. The energy released at each step of the chain is stored in a form of mitochondrion, can use to make ATP. Uh, that form ATP from ADP and in an organic phosphate. This mode of ATP synthesis is called oxidative phosphorylation. We call them oxidative phosphorylation because it is powered by the redox reaction of the electron transport chain. So in eukaryotic cells, the inner membrane of the mitochondria is the site of electron transport chain and another process called chemosmosis. Chemosmosis with the electron transport chain together making up oxidative phosphorylation. Oxidative phosphorylation accounts for the 90% of ATP generated by respiration. A smaller amount of ATP is formed directly in few reactions of glycolysis and citric acid cycle by a mechanism called substrate level phosphorylation. Some ATP is made by direct transfer of a phosphate group from an organic substrate to ADP by an enzyme to form ATP. This mode of ATP synthesis occurs when an enzyme transfers a phosphate group from a substrate molecule to ADP rather than adding an organic phosphate to ADP as in oxidative phosphorylation. The word glycolysis means sugar splitting, and that is exactly what happens during this pathway. Glucose, a 6-carbon sugar, is split into two of 3 carbon sugars. These smallest sugars are then oxidized and their remaining atoms rearranged to form 2 molecules of pyruvate. As summarized in this image, glycolysis can be divided into 2 phases, the energy investment phase and energy payoff phase. During the energy investment phase, the cell actually spends ATP. This investment is repaid with interest during the energy payoff phase. When ATP is produced by substrate level phosphorylation and NAD is reduced to NADH by electrons released from the oxidation of glucose, the net energy yield from glycolysis per glucose molecule is 2 ATP. plus 2 NADH. Uh, all of the carbon originally present in glucose is accounted for uh, in the two molecules of pyruvate. No carbon is released as carbon dioxide during glycolysis. Glycolysis occurs whether or not oxygen is present. However, if oxygen is present, the chemical energy stored in pyruvate and NADH can be extracted by pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation. So here in this uh, diagram, for the input and output from glycolysis, the energy investment phase, 2 ATP goes to 2 ADP and 2 in organic phosphate. Glucose enters the pathway uh, energy payoff phase, 4 ADP and 4 in organic phosphate goes to ATP, 2 NAD and 4 electrons plus 4 hydrogen ions goes to 2 of NADH and 2 hydrogen ion. Glucose is split into 2 pyruvate and 2 water, 2 molecules or 2 water molecules. And the net is uh, glucose goes to 2 pyruvate and 2 water molecules. 4 ATP formed minus 2 ATP used. That means we have only 2 ATP. 2 NAD and 4 electrons, 4 protons uh, goes to 2 NADH and 2 protons. With the glycolysis with more details, we will start with the energy investment phase. Here we have an enzyme that converts the glucose to glucose 6-phosphate. This enzyme is hexokinase. Anyway, it transfers ATP or phosphate group from ATP to glucose. And when ATP is used, going to ADP. With the enzyme hexokinase to form glucose 6-phosphate, which has the same structure as the glucose, except that uh, there is a phosphate group from ATP. Okay. In the second step, glucose 6-phosphate will convert to the fructose 6-phosphate, and then again in the third step, another an organic phosphate from the ATP will use to energize the fructose 6-phosphate and form fructose 1,6 biphosphate. Biphosphate means it contains two phosphate group, those two groups from the two ATP used in the first step and in the third step. Finally, the fructose 1,6 biphosphate split into two 3-carbon molecules called glyceraldehyde 3-phosphate, each of them with one phosphate group. We have two molecules from the investment phase. We have a glyceraldehyde 3-phosphate, two molecules of glyceraldehyde 3-phosphate from the first investment phase. Each of them will go through the second phase of energy payoff, and glyceraldehyde 3-phosphate will transfer to 1,3 by bisphosphate glyceraldehyde, glycerate, and 1,3 phosphoglycerate will transfer to 3-phosphoglycerate, and then 3-phosphoglycerate will transfer to 2-phosphoglycerate, and then to phosphoenol pyruvate, and then phosphoenol pyruvate transfer to pyruvate. So finally, we have 2 pyruvate or 2 molecules of pyruvate, and we have 2 energy or 2 ATP molecule produced in this phase, and 1 molecule of NADH by converting NAD to NADH and ADP to ATP. So the net is 2 ATP from this phase with 1 molecule of NADH. Glycolysis releases less than a quarter of the chemical energy in glucose that can be harvested by cells. Most of energy remains stockpiled in the 2 molecules of pyruvate. When oxygen is present, the pyruvate in eukaryotic cells enters the mitochondria, where the oxidation of glucose is completed. In aerobic respiration that occurs in prokaryotic cells, this process occurs in the cytosol. So upon entering the mitochondria, pyruvate is first converted to a compound called acetyl-CoA or coenzyme A. This step linking glycolysis and citric acid cycle is carried out by multi-enzymes, a complex that catalyze the reaction. Pyruvate carboxyl group, already somewhat oxidized and thus carrying a little chemical energy, is now fully oxidized and giving off a small molecule of carbon dioxide. So this is the first step in which carbon dioxide is released during respiration. The next step, the remaining 2 carbon fragment is oxidized and the electrons transfer to NAD and form storage energy in the form of NADH. Finally, coenzyme CoA, a sulfur-containing compound derived from vitamin B, is attached to sulfur atom to the 2 carbon intermediate forming acetyl-CoA. So this sulfur from the coenzyme A is attached with the acetyl-CoA to form this molecule. This molecule or acetyl-CoA has potential energy, which is used to transfer the acetyl group to a molecule in the citric acid cycle. This reaction that is a highly exergonic reaction. The citric acid cycle functions as metabolic furnace that further oxidize organic fuel derived from pyruvate. Pyruvate is broken down to 2,3 carbon dioxide molecules, including the molecule of carbon dioxide released during the conversion of pyruvate to acetyl-CoA. The cycle generates 1 ATP per turn by substrate level phosphorylation, but most of the chemical energy is transferred to NAD and FAD during the redox reaction. The reduced coenzymes NADH and FADH2 shuttle their cargo of high energy electrons into the electron transport chain. The citric acid cycle is also called the tricarboxylic acid cycle or the Krebs cycle. The Krebs cycle, this later in honoring Hans Krebs. The input and outputs per pyruvate molecule are shown with a focus on the carbon atoms involved. And to calculate one per glucose basis, multiply by 2 because each glucose molecule is split during glycolysis into 2 pyruvate molecules. So let's look at the citric acid cycle in more detail. The cycle has 8 steps, each catalyzed by specific enzymes. Acetyl-CoA from oxidation of pyruvate adds its 2-carbon acetyl group to oxaloacetate, producing citrate. Acetyl-CoA is a 2-carbon chain with both carbons shown in red so that they can be tracked throughout the cycle. So in the first step, acetyl-CoA from oxidation of pyruvate as its 2-carbon acetyl-CoA or acetyl group to oxaloacetate, producing citrate. And citrate is converted to its isomer called by removal one water molecule and addition of another, and it will form isostrate. And in the third step, isostrate is oxidized to alpha glutarate, reducing NAD plus to NADH. The resulting substrate loses carbon dioxide molecule. And then in fourth steps, alpha ketoglutarate will convert to succinyl, and another carbon dioxide is lost, and the resulting compound is oxidized, reducing NAD to NADH. And in fifth step, succinyl will convert to succinate, and it will produce ATP. And in the sixth step, 2 hydrogen are transferred to FAD and form FADH2, and oxidizing succinate to fumarate. And in seventh step, fumarate will convert to malate by adding of water molecule. And in the eighth step, malate will convert to oxaloacetate with the conversion of NAD to or reducing NAD to NADH. Glycolysis and citric acid cycle produce only 4 ATP molecules per glucose molecules, all by substrate level phosphorylation, 2 net ATP from glycolysis and 2 ATP from citric acid cycle. At this point, molecules of NADH and FADH2 account for most of the energy extracted from each glucose molecule. These electron scores link glycolysis and the citric acid cycle to the machinery of oxidative phosphorylation, which uses thereby released by the electron transport chain to power ATP synthesis. The electron transport chain is a collection of molecules embedded in the inner membrane of mitochondria in eukaryotic cells. The folding of inner membrane to form a crystal increases its surface area, providing space for thousands of copies of each component of the electron transport chain in mitochondria. Most components of the chain are proteins, which exist in multi-protein complexes numbered from 1 to 4, 1, 2, 3, 4. Highly bound to these proteins are processative group non-protein components such as cofactors and coenzymes, essential for the catalytic function of certain enzymes. Two electrons from NADH passes from NADH to complex 1. We have an electron transport chain. We have 4 complexes. We will say only complex 1, 2, 3, 4, and we have these two coenzyme Q and cytochrome C. So electron from NADH and 2 electrons from FADH2 will pass to this chain, electron transport chain. NADH will start with cytochrome or with the complex 1, while FADH2 will enter this chain starting with complex 2. This is the big difference between NADH and FADH2, and that's why the amount of energy with NADH is more than the energy produced by FADH2, since each of them will enter the chain in different with different sequence. Okay, NADH will start with complex 1, and FADH2 will start with complex 2. These 2 electrons from NADH or FADH2 move from the less electron-negative electron carrier, that means the complex 1, with the lower affinity for electron, to more electronegative electron carrier. This one is complex 4 in the chain. That releases free energy. The last electron carrier passes its electrons to oxygen, and the oxygen is very electronegative, and that's why oxygen will form water molecules. And remember that when electron is transferred or travel, it will travel electron with proton, that means in the form of hydrogen. Populating in the inner membrane of mitochondrion are many copies of protein complex called ATP synthase, which is the enzyme that makes up ATP from ADP and inorganic phosphate. ATP synthase works like an ion pumping running in reverse. Ion pumping usually use ATP as an energy source to transport ion against their gradients under the conditions of cellular respiration, rather than hydrolyzing ATP to pump proton against their concentration gradients. ATP synthase uses the energy of an existing ion gradient to power ATP synthesis. The power source for ATP synthase is a difference in the concentration of hydrogen, a pH difference on opposite sides of the inner mitochondrial membrane. This process, in which energy is stored in the form of hydrogen ion gradient across membrane, is used to drive cellular work such as the synthesis of ATP. So this process called chemosmosis. Hydrogen ion flowing down their gradient enter a channel in the stator, which is anchored in the membrane. Hydrogen ion are concentrated in the intermembrane space until they enter the stator. There is a clear concentration gradient of hydrogen ions across the mitochondrial membrane. Hydrogen ions enter binding sites within a rotor, changing the shape of each subunit so that the rotor spins within the membrane. Each hydrogen ion makes one complete turn before leaving the rotor and passing through a second channel in the stator into the mitochondrial matrix. Spinning of the rotor causes an internal rod to spin as well. This rod extends like a stalk into the knob below it, which is held stationary by part of the stator. Turning of the rod activates a catalyzed site in the knob that produce ATP from ADP and inorganic phosphate. How does the inner mitochondrial membrane generate and maintain the hydrogen gradient that drives ATP synthesis by the ATP synthase protein complex? Establishing the hydrogen gradient is a major function of the electron transport chain. The chain is an energy converter that uses exergonic flow of electrons from NADH and FADH2 to pump hydrogen across the membrane space. The hydrogen has a tendency to move back across the membrane, diffusing down its gradient, and the ATP syntheses are the only site that provides a route through the membrane for hydrogen. Uh, and the passage of hydrogen through ATP synthase uses the exergonic flow of hydrogen to drive the phosphorylation of ADP. Thus, the energy stored in hydrogen gradient across a membrane couples the redox reaction of electron transport chain to ATP synthesis. Certain combine of electron transport chain accept and release protons or hydrogen ion along with the electrons. At certain steps along the chain, electron transfer cause hydrogen to be taken up and released into the surrounding solution. The electron carriers are specially arranged in inner mitochondrial membrane in such a way that hydrogen is accepted from mitochondrial matrix and deposited in the intermembrane space. The hydrogen gradient that results is referred to as a proton motive force. And this emphasizing the capacity of the gradient to perform work. The force drives hydrogen ion back across the membrane through the hydrogen channels provided by ATP synthesis. In general terms, chemosmosis is an energy coupling mechanism that uses energy stored in the form of an hydrogen gradient across membrane to drive cellular work. NADH and FADH2 Several high-energy electron extracted from food during glycolysis and the citric acid cycle into electron transport chain built into the inner membrane of the mitochondrial membrane. The gold arrows trace the transport of electrons, which are finally passed through a terminal acceptor, which is the oxygen in the case of aerobic respiration, at the downhill end of the chain, okay, forming water. Most of the electron carriers of the chain are grouped into four complexes. So we have complex 1, 2, 3, 4, four complex. And we have two coenzyme. We have Q coenzyme and we have cytochrome C. So these two mobile carrier, epicnon Q and cytochrome C, move rapidly, flying electrons between the large complexes. As the complexes shuttle electrons, they pump protons from the mitochondrial matrix into the intermembrane space. FADH2 deposits its electron via complex 2 and so result in fewer protons being pumped into the membrane space than occurs with NADH. Chemical energy originally harvested from food is transformed into a proton motive force, a gradient of hydrogen across the membrane. During chemiosmosis, the protons flows back down their gradient via ATP synthase, which is built into the membrane nearby the complexes. The ATP synthase harness the proton motive force to phosphorylate ADP, forming ATP. Together, electron transport chain and chemosmosis make up oxidative phosphorylation. During respiration, most energy flows in this sequence: glucose, NADH, electron transport chain, and proton motive force, and then ATP. We can calculate the ATP profit when cellular respiration oxidizes a molecule of glucose to 6 molecules of carbon dioxide. The three main departments of this metabolic are glycolysis, pyruvate oxidation, and citric acid cycle, and the electron transport chain, which drives oxidative phosphorylation. In this figure, give a detailed counting of ATP yield for each glucose molecule that is oxidized. In the cytosol, glycolysis producing 2 ATP via substrate level phosphorylation, 2 pyruvate from glucose, which enters pyruvate oxidation inside the mitochondria, and 2 NADH, which carry electrons to oxidative phosphorylation inside the mitochondrion, and pyruvate oxidation to pyruvate from glycolysis are converted to acetyl-CoA, which will go to citric acid cycle. 2 NADH also carry electrons to oxidative phosphorylation in the citric acid cycle. 2 ATP are formed, and 6 NADH and 2 FADH2 carry electrons to oxidative phosphorylation. Oxidative phosphorylation produces about 26 to 28 ATP by oxidative phosphorylation, depending on which shuttle transport electrons from NADH in the cytosol. The maximum per glucose formation of ATP is about 30 to 32 ATP. Why are the numbers shown here inexact? There are three reasons we cannot state on exact number of ATP molecule generated by the breakdown of one molecule of glucose. The first one is phosphorylation and the redox reaction are not directly completed to each other. So the ratios of the number of NADH molecule to the number of ATP molecule is not a whole number. Each one NADH results in 10 hydrogen ion being transported out across the inner mitochondrial membrane. That generates enough proton motive force for the synthesis of 2.5 ATP. And the citric acid cycle also supplies electrons to the electron transport chain via FADH2 and NADH, but since it is electron enters later in the chain, the electron of FADH2 enter the chain later. It starts with the complex 2, not with the complex 1. Its electron enter later uh, with the complex 1. So each FADH2 responsible for the synthesis of 1.5 ATP. So each NADH give 2.5 ATP, and each FADH2 give 1.5 ATP. Second, the ATP yields varies slightly depending on the type of shuttle used to transport electrons from the cytosol into the mitochondrion. So depending on the kind of shuttle in a particular cell type, the electrons are passed either to NADA or FAD in the mitochondrial matrix. If the electrons are passed to FAD, only about 1.5 ATP can result from each NADH. That was originally generated in the cytosol. If the electrons are passed to mitochondrial NAD, the yield is about 2.5 ATP per NADH. The third reason is variable that reduces the yield is the use of the proton motive force generated by the redox reaction of respiration to drive other kind of work. If all the proton motive force generated by the electron transport chain were used to drive ATP synthesis, one glucose molecule could generate a maximum of 28 ATP produced by oxidative phosphorylation plus 4 ATP from substrate level phosphorylation to give a total yield of about 32 ATP, or only 30 ATP if the less efficient shuttle were functioning. So it depends on the shuttle that transferred the electron. However, there are two general mechanisms by which certain cells can oxidize the organic fuel and generate ATP without the use of oxygen, anaerobic respiration or fermentation. Uh, the distinction between these two type of respiration, fermentation and anaerobic, is that an electron transport chain is used in anaerobic respiration, but not in fermentation. Anaerobic respiration takes place in certain prokaryotic organisms that live in environments without oxygen. These organisms have an electron transport chain, but don't use electron, but don't use sorry, live in environment without oxygen. Those organisms, uh, Those have an electron transport chain but don't use oxygen as the final electron acceptor at the end of the chain, but instead use other substances such as sulfate ion at the end of their respiratory chain. Fermentation is a way of harvesting chemical energy without using either oxygen or any electron transport chain, in other words, without cellular respiration. How can food be oxidized without cellular respiration? As an alternative to respiratory oxidation of organic nutrients, fermentation is an extension of glycolysis that allows continuous generation of ATP by the substrate level phosphorylation of glycolysis. Fermentation consists of glycolysis plus reaction that generate NAD by transferring electron from NADH to pyruvate. The NAD can then be reused to oxidize sugar by glycolysis. There are many types of fermentation, differing in the end product formed from pyruvate. Two common types will be discussed here. These are alcohol fermentation and lactic acid fermentation. In alcohol fermentation, pyruvate is converted to ethanol, ethyl alcohol, in two steps. The first step is release oxygen. This step, the first step is release oxygen. Uh, uh, I'm sorry, the first step is release carbon dioxide from the pyruvate. From each pyruvate will release carbon dioxide, which is converted to two carbon compound called acetaldehyde. In the second step, acetaldehyde is reduced by NADH to ethanol. This regenerates the supply of NAD needed for the continuation of glycolysis. Many bacteria carry out alcohol fermentation under aerobic respiration conditions, anaerobic respiration. Yeast or fungus used alcohol fermentation in addition to aerobic respiration. For thousands of years, humans use, have used yeast in brewing, wine making, and baking. The carbon dioxide bubbles generated by baker's yeast during alcohol fermentation allow bread to rise. In lactic acid fermentation, during this fermentation, pyruvate from the glycolysis is released directly by NADH to form lactate as an end product, regenerating NAD with no release of carbon dioxide. Lactate is the ionized form of lactic acid. Lactic acid fermentation by certain fungi and bacteria is used in the dairy industry to make cheese and yogurt. Human muscle cells use lactic acid fermentation to generate ATP when oxygen is scarce. Some organisms called obligate anaerobies, which carry out only fermentation or anaerobic respiration. In fact, these organisms cannot survive in the presence of oxygen, some forms of which can actually be toxic if protective systems are not present in the cell. Obligate aerobies carry out only aerobic respiration and cannot survive in the absence of oxygen. Other organisms, including yeast and many bacteria, can make enough ATP to survive using either fermentation or respiration. such species called facultative anaerobies. In yeast cells, for example, pyruvate is a fork in the metabolic road that lead to two alternative catabolic roads. Under aerobic conditions, pyruvate can be converted to acetyl-CoA and oxidation continues in citric acid cycle via aerobic respiration. Under anaerobic conditions, lactic acid fermentation occurs. In this section, we will see that glycolysis and citric acid cycle are major intersection of the cell's catabolic breakdown pathway and anabolic or biosynthetic pathways. We use glucose as an example of a fuel for cellular respiration, but free glucose molecules are not common in the diet of human and other animals. We obtain most of our calories in the form of fats, proteins, and carbohydrates, such as sucrose and other disaccharides and starch. Starch is a polysaccharide. All these organic molecules in food can be used by cellular respiration to make ATP. Proteins are broken down into amino acids, which must then have an amino group removed. This amino removed from before entry into the cellular respiration pathway as a pyruvate during glycolysis or as acetyl-CoA or directly enter the citric acid cycle. Carbohydrates are broken down into sugars which can then enter glycolysis. Fats are broken down into glycerol and fatty acids. Glycerol can enter glycolysis as glyceraldehyde, 3-phosphate. So glycerol can enter the glycolysis as glyceraldehyde 3-phosphate. Fatty acids enter the citric acid cycle as acetyl-CoA. Cells need substances as well as energy. Not all the organic molecules of food are destined to be oxidized as a fuel to make ATP. In addition to calories, food must also provide the carbon skeleton that cells require to make their own molecules. Some organic monomers obtained from digestion can be used directly. For example, amino acid from the hydrolysis of protein in food can be incorporated into organisms' own proteins. However, our body needs specific molecules that are not present in food. Compounds formed as intermediates of glycolysis and the citric acid cycle can be directed into anabolic pathways as precursors from which the cell can synthesize the molecule it requires. For example, humans can make about half of the 20 amino acids in proteins by modifying compounds from the citric acid cycle. The cell doesn't waste energy making more of particular substances than it needs. If there is a surplus of certain amino acids, for example, the anabolic pathway that synthesizes the amino acids from intermediate molecules of the citric acid cycle is switched off. The most common mechanisms for this control is feedback inhibition. The end product of the anabolic pathways inhibits the enzyme that catalyzes an early step of the pathway. This prevents the needless diversion of key metabolic intermediates from uses that are more urgent. The cell also controls its catabolism. If the cell is working hard and its ATP concentrations begin to drop, cellular respiration speed up. When there is plenty of ATP to meet demand, respiration slows down. Uh, so controls is based mainly on regulating the activity of enzymes at a strategic point in the catabolic pathway. During glycolysis, glucose is converted to fructose 6-phosphate and then to fructose 1,6 uh, biphosphate with the allosteric enzyme phosphofructokinase. Okay. After a series of reactions, fructose 1,6 biphosphate is converted to pyruvate, forming ATP. In the process, pyruvate is converted to acetyl-CoA, which enters the citric acid cycle, the product of which go on to oxidative phosphorylation. Both of these steps also result in the formation of ATP. Both ATP and citrate produced uh, during the citric acid cycle inhibits phosphofructokinase. Those ATP will inhibit the phosphofructokinase, preventing progression of the pathway. AMP, also derived from ADP, stimulates the phosphofructokinase.